Hello everyone, this is Dr. P. Mary Anupama, Assistant Professor and Head of the Department of Biochemistry, St. Joseph's College for Women. Through this video, I'll be discussing the topic Glycogenesis. The topics that will be covered include basic introduction to what is glycogenesis, where it takes place, that is the location of glycogenesis, the steps of glycogenesis in detail, and then we have regulation as well as importance of glycogenesis. All these will be dealt in this video. So what is glycogenesis? Glycogenesis is the biosynthesis of glycogen. We know that glycogen is a branched polymer of glucose and it often serves as the storage form of carbohydrate in animals. And this process, which converts the excess glucose into the storage form of glycogen, it is seen primarily in liver and skeletal muscles. So there, the excess glucose is converted to glycogen, and this is what it is, glycogenesis. Glycogenesis is defined as the metabolic pathway through which glucose molecules are converted into glycogen and then further stored. It occurs when glucose levels are very high, which means after a meal or when you have carbohydrate rich breakfast, then the glucose, it is taken and then it is stored as energy resource for later use. The sites of glycogenesis. There are two sites of glycogenesis. We have the liver and the skeletal muscles. We all know that liver, it stores glycogen, and this is mainly for maintaining the blood glucose levels between meals and also when you are fasting. So here the glycogen, when it is broken down, it releases glucose into the blood. This is because the liver has this enzyme glucose 6 phosphatase. Next is skeletal muscles. Skeletal muscles, they also store glycogen for local use when muscle contraction is taking place. So this particular skeletal muscles, they lack glucose 6 phosphatase. So they do not contribute to blood glucose levels, but they are involved in glycogen synthesis and storage. Now let us see the steps of glycogenesis. Glycogenesis involves many enzymatic steps and it takes place in cytosol. And this glycogenesis requires ATP, UTP besides glucose. Synthesis of UDP glucose. So here the enzyme hexokinase, which is present in muscles, and glucokinase, which is present in the liver, they convert glucose to glucose 6 phosphate. Then we have phosphoglucomutase, which catalyzes the conversion of glucose 6 phosphate to glucose 1 phosphate. Later on, uridine diphosphate glucose, it is synthesized from glucose 1 phosphate and UTP by UDP glucose pyrophosphorylase. So all these, starting from glucose to glucose 6 phosphate, glucose 6 phosphate to glucose 1 phosphate, then glucose 1 phosphate to UDP glucose formation, it involves the first step. The second step in glycogenesis is requirement of primer to initiate glycogenesis. So here you have a small fragment of already existing glycogen that must act as a primer to initiate the synthesis of glycogen. So you have here glycogen initiator synthase that transfers one molecule of glucose to this glycogenin. So you have UDP glucose, which is produced in the previous step, that is transferred on to glycogenin. So then glycogen in itself, it takes up few more glucose moieties to form a fragment of a primer. So you have the glycogen primer which is produced. Now this is ready to accept some more glucose molecules. The third step is glycogen synthesis by glycogen synthase. So glycogen synthase, it is primarily responsible for the formation of the alpha 1,4 glycosidic bonds between the glucose molecules. So we all know that UDP glucose will be contributing to the glucose molecules, and with time the glycogen synthase keeps on adding these molecules, and you see that alpha 1,4 linkages are formed by this glycogen synthase between the glucose molecules. The fourth step is the formation of branches in glycogen. So here once glycogen synthase it is acting upon the primer, it is resulting in the formation of linear and unbranched molecule, and this has only alpha 1,4 glycosidic linkages. But we all know that glycogen is like a branched tree, and the formation of branches, it requires a specialized enzyme. We call it as the branching enzyme. So here this branching enzyme is called as the glycosyl alpha 4,6 transferase, or you also call it as amylo alpha 1,4,1,6 trans glucosidase. So this enzyme is mainly responsible for the branching. So it transfers around 5 to 8 glucose residues from the non-reducing end of glycogen chain by breaking the alpha 1,4 linkages to another glucose residue where it is linked by alpha 1,6 bond. This leads to formation of new non-reducing ends besides the existing ones. So here you have non-reducing ends which are formed. Glycogen is further elongated and branched respectively by this by the enzyme glycogen synthase. So glycogen synthase will be adding new moieties over here, and again the other enzyme, that is glucosyl 4,6 transferase, will be involved in branching process. This is how glycogen molecule is synthesized and polymerized step by step. So the overall reaction of glycogen synthesis for the addition of each glucose residue, it is like this. So you have the glucose and molecules. To add one molecule of glucose to it, it requires 2 ATP molecules, and finally you will be getting glucose N plus 1, resulting in the formation of 2 ADP and PI. So this ATP, out of these two, one, it is used for the phosphorylation of glucose where you get glucose 6-phosphate, while the other one is used for the conversion of UDP to UTP. Regulation of glycogenesis. So we all know that glycogenesis, it is regulated by hormones as well as by allosterically regulated enzymes. So here first is stimulation. The glycogenesis pathway, it is stimulated by the hormone insulin, wherein insulin, it activates glycogen synthase through dephosphorylation. So when does insulin come? When glucose levels are high, which is required for the synthesis of glycogen. So whatever blood glucose is there, it has to be converted to glycogen. So glycogen synthase enzyme is activated. So when you have high glucose 6-phosphate also, there is allosteric activation of glycogen synthase which is taking place. Coming to inhibition. Glucagon is the hormone which is regulating it, as well as the epinephrine which is seen in the muscles as well as liver. So both these things will be regulating. They activate the protein kinases which inactivate glycogen synthase. That is how glycogenesis is regulated. So how is glycogenesis important to us? First thing is, It prevents hyperglycemia condition which we may have to face after meals because we eat lot of carbohydrate based food and lots of glucose will be released. So here that excess glucose is stored by glycogenesis. It also maintains the glucose supply during fasting via glycogenolysis. If glycogen is stored, then only glycogenolysis will be taking place. That is, glycogen hydrolysis will be taking place and glucose is released. Next one is muscle glycogen. Yes, it provides very quick energy when a person is exercising. So this is the importance, the three important points that we have to remember why glycogenesis is required. So we have covered the topics, the basic introduction to glycogenesis, followed by its localization, where it takes place, and the steps in detail, followed by regulation as well as importance. The following textbooks are referred while preparing this video. It includes Biochemistry, The Principles of Biochemistry by Leninger, Biochemistry by U. Satyanarayana, as well as Harper's Biochemistry. Thank you.